Welcome, curious minds, to another deep dive. What if I told you that the very blood test you might get before surgery uses technology that's also helping us fight cancer at a genetic level?
It's pretty amazing how interconnected these things are.
Totally. Today, we're diving deep into the powerful toolkit immunologists use every single day. From generating custom antibodies to editing genes and even watching cells move inside a living organism. These are the methods that allow us to understand, diagnose, and even treat diseases at a really fundamental level.
Absolutely. It's the engine room of modern immunology in many ways.
And our mission for you, our listener, is really to gain a master-level conceptual understanding of these experimental systems. We want you to grasp the core principles, understand why certain choices are made in experiments.
Right, the thinking behind the method.
Exactly. And evaluate findings more critically, maybe even imagine how you might design your own investigations. It's about getting a feel for how the elite professionals in this field operate, without getting bogged down in every tiny protocol step.
That's a great goal. And it's true, modern immunology, while it borrows heavily from diverse fields like, structural biology, physics, biochemistry, computer science, it has in turn given back this immense arsenal of tools to the entire scientific community.
And a lot of those tools revolve around antibodies, right?
They really do. Many of these foundational methods revolve around the very molecules that are central to immunity, antibodies.
Here's where it gets really interesting for me. Antibodies, these amazing proteins, they're truly at the heart of so much of what we do in immunology. They don't just fight disease in our bodies. They are absolutely invaluable tools for understanding it in the lab.
Yeah.
It's immunology's unique contribution, you could say.
I think that's fair. What's truly fascinating here is how the exquisite specificity and binding properties of antibodies make them unparalleled tools. Imagine a microscopic key that only fits one very particular lock, or maybe a few very similar ones.
Yeah.
That's essentially an antibody. This incredible precision allows immunologists to detect specific molecules, purify them from really complex mixtures, and even visualize them within cells or tissues with fantastic clarity. This capability fundamentally changed how we study biological systems.
And it all starts with actually getting these keys. How do we generate them? I know there are a couple of main ways. One more natural, one more engineered.
That's a good way to put it. There are two main approaches. Polyclonal antibodies, which mimic the body's natural diverse response, and monoclonal antibodies, which are highly engineered for incredible precision. Let's start with the polyclonal one.
Okay, polyclonal. Sounds like many clones.
Exactly. Polyclonal antibodies are essentially the body's natural immune response, harvested for our use. When you immunize an animal, say a rabbit or a goat, with an antigen, like a virus or a specific protein, their B cells get activated.
Right.
But not just one B cell. Multiple different B cell clones will respond, and each clone produces antibodies that target slightly different parts, different epitopes, on that single antigen molecule.
So you get a mix of antibodies hitting different spots on the target.
Precisely. Because these antibodies come from many different B cell clones targeting different parts of the antigen, they're called polyclonal.
So how do we actually obtain them? Do we just inject something and wait?
Pretty much, yeah. You immunize an experimental animal with the antigen, but to really boost that immune response, we often include substances called adjuvants.
Ah, adjuvants. I've heard that term, especially with vaccines. What do they do exactly?
Adjuvants are like the immune system's alarm bell. They're substances added to the immunizing preparation to essentially make the body pay more attention, boosting the response by deliberately engaging the innate immune system.
The first line of defense.
Right. This helps activate both the antigen-specific B cells and T cells more effectively.
Okay. Are there different kinds?
Oh, yes. Traditionally, some adjuvants, like Franz, were quite potent. Franz's complete adjuvant actually included dead mycobacteria to really ramp up the innate response.
Sounds intense.
It was, and it worked, but it also caused significant local inflammation, sometimes even tissue damage. So those are now largely avoided due to ethical concerns for animal welfare.
Good.
Modern adjuvants are much more sophisticated. They leverage our understanding of how the innate immune system gets triggered. Some mimic viral RNA, for example, basically tricking the immune system into thinking there's a viral infection.
Which kicks off a strong response.
Exactly. Others, like ELIM, which is commonly used in human vaccines like the HPV or Hep B vaccines, are thought to work differently, maybe by creating a slow-release depot for the antigen, giving the immune system sustained exposure, or perhaps enhancing uptake by immune cells. The exact mechanism for alum is still debated, surprisingly.
Interesting. So polyclonal antibodies, this mix targeting different epitopes.
Yeah.
What are they actually good for in the lab?
Their key advantage lies in their ability to cross-link multiple antigen molecules really effectively. Because they target various epitopes, they can form these large, intricate antibody complexes or lattices.
Like building a bridge between antigens.
Yeah, sort of, yes. This property is particularly useful for techniques like agglutination clumping or immunoprecipitation, which rely on these large complexes forming and becoming visible or easily separable. They essentially cast a wide net.
Okay, a wide net. But I imagine that wide net comes with some drawbacks, especially if you need, pinpoint accuracy.
It absolutely does. The main disadvantages of polyclonal preparations are twofold. First, they can exhibit cross-reactivities. Some antibodies in that mix might bind not only to your intended target, but also to related, or sometimes even seemingly unrelated, antigens. That can give you false positives or confusing results.
Right, binding to the wrong thing.
Exactly. Second, there's significant variability. The antibodies you get from one bleed or blood drawing might differ from the next one, even from the same animal. The immune response matures over time, the affinity changes. It's not perfectly reproducible.
So if you're running a diagnostic test, that variability would be a huge problem, right? You need consistency.
Precisely. That lack of reproducibility makes polyclonal antibodies less suitable for highly quantitative or standardized diagnostic or research applications where consistency is absolutely paramount.
Okay, so if polyclonals are the wide net, we need a laser-guided missile for precision. And that brings us to monoclonal antibodies, or mAbs. This was a massive breakthrough, wasn't it?
Oh, absolutely. A game changer. The ability to generate these precise tools was discovered back in 1975 by Georges Kohler and Cesar Milstein. It was Nobel Prize-winning work.
Because it solved those problems of variability and cross-reactivity.
Exactly. The disadvantages of polyclonal antibodies are almost entirely eliminated with monoclonal antibodies because, by definition, a mAb is the product of a single stimulated B cell clone. Its binding site is identical, it never changes.
One B cell, one antibody type forever. How on earth did they manage that? You can't just keep one B cell alive indefinitely, can you?
Not normally, no. That was the genius of their hybridoma technology. The core concept is brilliant. You take a normal activated antibody-producing B cell, which has a short lifespan in a lab dish.
Right.
And you fuse it with a myeloma cell. Myeloma cells are cancerous plasma cells, and crucially, they can grow indefinitely in culture. They're immortal.
So you fuse a mortal antibody factory with an immortal cell.
Precisely. The resulting fused cell is called a hybridoma. It inherits the immortal growth property from the myeloma parent and critically continues to secrete the unique, specific antibody produced by the original B cell parent.
Wow.
And to make it even cleaner, they engineered the myeloma cells used for fusion so they don't synthesize their own antibodies. This ensures that the hybridoma only produces the single desired antibody from the B cell partner.
Okay, that's clever. So you're creating an immortal antibody factory from a single B cell. How do you actually get them to fuse? And then how do you pick out the successful hybrids from all the other cells floating around the unfused B cells, the unfused myelomas?
Good questions. Fusion is typically done using chemical agents like polyethylene glycol or PEG, which helps cell membranes merge. But after fusion, you have this messy mixture. Unfused B cells, which die off naturally pretty quickly.
Okay, they're taken care of.
Unfused myeloma cells, which are immortal and would otherwise take over the culture. And myeloma hybrids, also immortal and unwanted. And then your desired B cell myeloma hybridomas, so you need a selection strategy.
Right. How do you kill off the immortal myeloma cells but keep the immortal hybridomas?
This is where the HAT medium comes in. It's a really elegant piece of biology. The myeloma cells used are specifically chosen because they have a genetic defect. They lack an enzyme called HGPRT. This means they can't use a certain salvage pathway for making DNA.
Right.
Normal cells, including B cells, have functional HGPRT and can use this salvage pathway. The HET medium contains a drug called aminopterin, which blocks the main DNA synthesis pathway, the de novo pathway.
So you block the main road.
Exactly. Now, all cells have to rely on the salvage pathway, but the myeloma cells can't use it because they lack HGPRT. So unfused myeloma cells and myeloma hybrids just die in HET medium.
But the B cells have HGPRT.
Right. And when a B cell fuses with the myeloma cell, the resulting hybridoma gets the functional HGPRT from the B cell parent. So the hybridoma can use the salvage pathway and survive and proliferate in the HNT medium while all the unwanted cells die off.
That is incredibly clever. You're essentially designing a genetic trap that only the successful hybridomas can escape, turning a biological quirk into a powerful selection tool.
It really is. After selection and some stabilization, sometimes hybridomas initially shed some chromosomes. These hybridoma cell lines can be cultured indefinitely. They become reliable factories, secreting large quantities of identical monoclonal antibodies with a completely defined specificity and known cross-reactivity.
So consistent, specific, reproducible, ideal for diagnostics.
Absolutely. The specificity, affinity, and any minor cross-reactivity of mAbs are entirely stable over time. This makes them perfect for diagnostic tests like tissue typing, identifying specific disease biomarkers, or for research where you need that absolute consistency run after run.
Are there any downsides compared to polyclonals?
Just a minor one, really. For certain applications like agglutination, where you want really big clumps, polyclonals can sometimes be better. Because they bind multiple epitopes, they can cross-link antigens more extensively. Monoclonals, binding only one epitope type, form smaller complexes. But for the vast majority of applications where specificity is key, mAbs are the clear winner.
And the power of mAbs doesn't stop there, right? Once you have this highly specific key, scientists have figured out ways to modify them, customize them for even more diverse jobs.
That's absolutely right. Monoclonal antibodies provide this wonderfully reproducible binding site for almost any target imaginable. And researchers have been incredibly imaginative in leveraging this. They can be genetically altered, for instance.
Like making them more human.
Exactly. A common manipulation is creating chimeric antibodies. You essentially take the mouse antibodies binding sites, the variable regions that recognize the target, and genetically graft them onto the constant regions of a human antibody.
Why do that?
It's crucial for therapies. If you inject a pure mouse antibody into a human, the human immune system sees it as foreign and attacks it, causing inflammation and reducing effectiveness. By humanizing the antibody, making most of it human protein, you drastically reduce this immune reaction. These are essential for many antibody-based drugs.
Makes sense. What else can you do? Stick toxins on them.
Yes, precisely. Some antibodies are modified by conjugation to toxins or potent drugs. These are like guided missiles. The antibody targets a specific cell type, like a cancer cell, and delivers a lethal payload directly to that cell, minimizing damage to healthy tissues. There are several successful antibody-drug conjugates, or ADCs, used to treat certain breast cancers and leukemias, for example.
It's like taking a finely tuned instrument and then adding all sorts of cool attachments to make it even more powerful for specific jobs.
That's a perfect analogy. Antibodies can also be chemically linked, conjugated to other molecules without messing up their antigen binding. For diagnostic tests like ELISAs, which we'll get to, they can be linked to enzymes or a molecule called biotin. For microscopy or cell sorting, they're often tagged with fluorescent dyes or tiny synthetic beads. There are even attempts to make Abzymes antibodies engineered to act like enzymes themselves, though that's more specialized.
What about T cells? Can you make hybridomas from them too?
You can, yes. T-cell hybridomas have been generated with defined specificities. They've been really useful for studying T-cell receptor biochemistry, cytokine secretion patterns, and T-cell subpopulations. However, they're less useful therapeutically or diagnostically compared to B-cell hybridomas because T-cells don't secrete a soluble antigen-binding molecule like antibodies do. Their recognition is much more complex, involving MHC molecules presenting peptides.
Right, much harder to use as a simple regen. Okay, so we have these incredible custom-built antibodies. Now, how do we actually use them to detect and quantify things in biological samples? Let's talk about antibody-based assays, starting with the classics, immunoprecipitation and agglutination.
Sounds good. The core principle behind both immunoprecipitation and agglutination is that antibodies are typically multivalent, usually bivalent, meaning they have two antigen binding sites. This multivalency allows them to bridge or cross-link multiple antigen molecules, forming these large, often visible antigen-antibody complexes. It's this complex formation that these techniques exploit.
All right, so immunoprecipitation, IP. It sounds like you're literally pulling something out of a solution using an antibody.
That's exactly what it is in its simplest form. Immunoprecipitation in solution involves mixing antibodies with soluble antigens. If the conditions are right, roughly equivalent amounts of antibody and antigen, the antibodies cross-link the antigens into large, insoluble complexes that precipitate or fall out of solution.
And why would you do that?
It's a classic method to purify specific soluble molecules from a complex mixture, like serum. Historically, it was crucial. Elvin Kabat used it to definitively characterize antibodies themselves as belonging to the gamma globulin fraction of serum proteins. You can also use it to specifically remove an antigen from a solution.
So it's a purification or depletion method.
Yeah.
What about doing this in gels? Is that different?
It's the same principle, but visualized differently. Immunoprecipitation in gel matrices, often using the Outreloni method or double immunodiffusion, involves letting antigen and antibody diffuse towards each other from separate wells cut into an agar gel.
Okay.
Where they meet at optimal concentrations, the zone of equivalence they form, that precipitate, but this time it's trapped in the gel matrix as a visible white line, a precipitin line.
What's the advantage of doing it in a gel?
It's simple, relatively rapid, and visually intuitive. It's still used today for detecting the presence of certain antibodies or antigens, and importantly, for determining cross-reactivity patterns. By arranging wells with different antigens and antibodies, you can see if two antigens are recognized by the same antibody pool, indicating they share epitopes.
So it tells you about relationships between antigens. Now, you mentioned earlier that IP is key for pulling molecules out of messy samples. How does that work with, say, cell extracts?
Right. IP is heavily used to isolate specific protein antigens directly from complex cell and tissue extracts. Typically, you'd lyse the cells using detergents to solubilize the proteins. Then you add your specific antibody, the primary antibody, to this extract.
How do you get the antibody-antigen complex out easily?
Good question. You usually add a secondary reagent that binds the primary antibody. This secondary reagent is often pre-attached to a solid phase support, like synthetic beads or, very commonly now, magnetic beads.
Ah, so you can just use a magnet to pull everything out.
Exactly. You add the magnetic beads coated with, say, protein A or protein G, which bind strongly to antibody Fc regions, incubate, then use a magnet to pull down the beads, which now have the antibody-antigen complexes attached. You wash away everything else, then elute your target protein off the beads.
Clever. And then how do you know you actually got the protein you wanted, or if
Yeah.
anything else came along for the ride?
That's where downstream analysis comes in, most commonly SDS page followed by Western blotting. You run your precipitated proteins on a gel to separate them by size, then use another antibody on the Western blot to specifically detect your protein of interest. This confirms you pulled it down, gives you an estimate of its abundance, and crucially can identify other proteins that co-immunoprecipitated with it.
Meaning they were stuck to your target protein inside the cell.
Precisely. This co-IP approach was fundamental in discovering that receptors like the T-cell receptor and B-cell receptor weren't single proteins, but actually existed as part of multi-protein complexes, revealing their co-receptors and signaling partners.
Okay, so immunoprecipitation is about pulling things out. Now, what about agglutination? It sounds like clumping, which ties back to those large complexes you mentioned.
Exactly. Agglutination works on the same principle of antibody-mediated cross-linking, but it involves particulate antigens, usually whole cells, rather than soluble molecules. When antibodies bind to antigens on cell surfaces, they cross-link the cells themselves, forming large aggregates or clumps.
And because cells are much bigger than molecules, these clumps are easy to see.
Right. The aggregated product is often large enough to be seen with the naked eye, making it a very direct and simple readout.
The classic example that always comes to mind is blood typing, right? That little card where they mix blood drops with different sera.
That's hemagglutination, agglutination of red blood cells, erythrocytes, and it's a perfect example. Antibodies specific for ABO blood group antigens on the red blood cells will cause them to visibly clump together if the antigen is present. It's routinely performed for blood typing before transfusions, making it one of the world's most frequently used immunoassays because it's easy, rapid, and doesn't require sophisticated instruments.
It's amazing how fundamental that is. Now, what about hemagglutination inhibition? That sounds like you're stopping the clumping instead.
Yes, it's a clever twist used mainly in virology. Some viruses, notably influenza virus, have surface proteins like influenza's hemagglutinin or HA protein that naturally bind to molecules on the surface of red blood cells and cause them to agglutinate all by themselves.
Okay, the virus itself causes clumping.
Right. The hemagglutination inhibition, or HI, assay leverages this. It's used to detect the presence of antibodies against the virus in a patient's serum. You mix the patient's serum with the virus first. If the serum contains antiviral antibodies, like anti-HA antibodies, they'll bind to the virus and block its ability to agglutinate the red blood cells you add later.
So no clumping means the patient have antibodies.
Exactly. The more antibodies the patient has, the more you can dilute their serum, and it will still inhibit the virus-induced hemagglutination. It gives you a titer, a measure of the antibody level. It's incredibly useful for diagnosing viral infections and tracking the immune response to specific strains, like different flu strains.
And does this work for bacteria too?
Yes, the same principle applies in bacterial agglutination reactions. Antibodies against bacterial surface antigens will cross-link the bacteria, causing them to form visible clumps or pellets. This is used to diagnose certain bacterial infections, like typhoid fever, by measuring the agglutinin titer in patient serum, and also for typing different bacterial species in the lab.
All right, so we've covered these classic, often visual clumping and precipitation methods. Now let's move into what sound like more modern, high-throughput approaches. Solid phase support assays. This is where automation really kicked in, right?
Yes, these assays were a major leap forward in terms of sensitivity, speed, and automation. The key idea is binding either the antibody or the antigen to a solid surface, like the wells of a microtiter plate or tiny beads. This makes washing steps much easier and allows for processing many samples simultaneously, often robotically.
And the first big one here was the radioimmunoassay, or RIA.
That's right. RIAs really revolutionized biological measurement. Before RIAs, developed in the 1950s by Solomon Bersin and Rosalind Yaloyello, later won the Nobel for it, many biologically important molecules like hormones or cytokines were present in such tiny concentrations in body fluids that they were simply undetectable.
Too low to measure.
Exactly. RIAs made it possible to measure substances down to the picogram range. That's a trillionth of a gram. It was incredibly sensitive for its time.
And did it work? Using radioactivity, obviously.
Yes. The most common format is a competitive RIA. You typically coat a surface, like a well, with an antibody specific for the molecule you want to measure. Then you add your sample, which contains an unknown amount of the unlabeled molecule, or antigen. Crucially, you also add a known constant amount of the same antigen that has been radioactively labeled, usually with iodine-125.
So the labeled and unlabeled antigen compete for the antibody.
Precisely. The unlabeled antigen from your sample competes with the labeled antigen for binding to the limited number of antibody sites on the well. The more unlabeled antigen there is in your sample, the less of the radioactive labeled antigen will be able to bind.
So higher sample concentration means lower radioactivity bound to the well.
Exactly. You wash away the unbound stuff, measure the radioactivity remaining in the well using a gamma counter, and compare that to a standard curve generated using known concentrations of unlabeled antigen. This allows you to determine the concentration in your unknown sample.
Brilliant, but I guess the radioactivity was the downside.
It was. While incredibly sensitive, RIAs generated significant amounts of radioactive waste, and there were safety concerns for the scientists working with isotopes. This really spurred the development of safer, non-radioactive alternatives, which brings us to the workhorse of many labs today, enzyme-linked immunosorbent assays, or ELISAs.
Ah, ELISAs.
Yes.
You see those mentioned everywhere.
Oh.
How do they avoid the radioactivity?
The core principle is very similar to RIA, but instead of a radioisotope, ELISAs use enzymes covalently linked to an antibody. These enzymes act on a specific substrate, converting it into a product that can be easily measured. Usually it's a colored product detected by absorbance, but it can also be fluorescent or chemiluminescent, light-producing.
So enzyme activity replaces radioactivity count.
Exactly. And the beauty is that ELISAs can match the sensitivity of RIAs, but they're much safer, often less costly, and the reagents generally have a longer shelf life.
Are there different kinds of ELISA?
Yes, several common variations exist, used either qualitatively, is the target present, or quantitatively, how much is present. The indirect ELISA is typically used to detect specific antibodies in a sample, like serum. You coat the wells with the antigen your antibody binds to, then add the sample, for example, patient serum. If the specific antibody is present, it bonds. Then you add a secondary antibody that recognizes human antibodies, or whatever species your primary antibody is from, and is linked to an enzyme. Wash, add substrate, and measure the color change. This is how standard HIV antibody tests work, for instance.
Okay, so indirect detects antibody. What about detecting the antigen itself?
For that, the most common format is the sandwich ELISA. Here, you start by coating the well with a capture antibody that's specific for your antigen. Then you add your sample. If the antigen is present, it gets captured by the antibody on the well surface. After washing, you add a second detection antibody, which is also specific for the antigen but binds to a different epitope than the capture antibody. This detection antibody is linked to an enzyme. Wash again, add substrate, and measure the signal. The amount of signal is directly proportional to the amount of antigens sandwiched between the two antibodies.
Requires two different antibodies for the same target.
Yes, that's key for the sandwich format. It needs two antibodies that can bind the antigen simultaneously without interfering with each other. This is widely used for measuring soluble molecules like cytokines in culture supernatants or serum. There's also a competitive ELISA, which, like competitive RIA, measures antigen based on its ability to compete with a labeled antigen for antibody binding, resulting in an inverse relationship between antigen concentration and signal.
These sound incredibly versatile. Are there ways to make them even more sensitive or flexible?
Definitely. One very common trick to boost sensitivity involves using biotin and streptavidin. Biotin is a small vitamin that binds to the protein streptavidin with extraordinarily high affinity, one of the strongest non-covalent bonds known in nature.
How does that help in ELISA?
You can conjugate your primary or secondary antibody to biotin. Then, instead of using an enzyme-conjugated antibody directly, you use streptavidin that's conjugated to the enzyme. Since streptavidin has 4 binding sites for biotin, and you can often attach multiple biotins to an antibody, you can potentially bind multiple enzyme-linked streptavidin molecules per antibody complex. This amplifies the signal significantly because each enzyme molecule can turn over many substrate molecules. It's a powerful amplification strategy.
Clever. Any other ELISA twists?
Yes, a relatively newer development is the in-cell ELISA. This allows you to measure intracellular proteins without actually breaking open the cells. You grow the cells directly in the wells, fix and permeabilize them, then perform the antibody and detection steps right there. It avoids potential issues with proteins degrading or precipitating during extraction and allows for automated quantitative analysis while keeping the cells somewhat intact.
That's neat. Now, moving from measuring the amount of a molecule in a sample to actually counting how many individual cells are producing it, that seems like a big jump in information. That's what ELISpot assays are for, right?
Exactly. This is a really fascinating technique because it shifts the focus from bulk measurement to single-cell function. ELISpot, which is a modification of the sandwich ELISA principle, quantitatively determines the number of cells within a population that are actively secreting a specific molecule, like a cytokine or an antibody.
How does it work? How do you see individual cells?
You start with a plate coated with a capture antibody for the molecule of interest, say interferon gamma, IFN. You then add a known number of cells to the well and stimulate them if necessary. As the cells settle, any cell secreting IFN-nu will release it, and it gets immediately captured by the antibodies coated on the plate surface right around that cell.
Forming a little halo of captured cytokine.
Essentially, yes. You then wash away the cells. Now you add a detection antibody for IFN-F linked to an enzyme, just like in a sandwich ELISA. Wash again. Finally, you add a special ELISpot substrate. This substrate, when acted upon by the enzyme, forms a colored, insoluble precipitate.
Oh, so it makes a solid spot instead of coloring the whole solution.
Precisely. You end up with a distinct colored spot on the membrane at the location where each secreting cell originally sat. You can then count these spots, either manually under a microscope or using an automated reader. Knowing the number of spots and the initial number of cells plated, you can calculate the frequency of cytokine-secreting cells in your population. It's a very sensitive way to measure cellular immune responses.
That provides a powerful functional readout at the single-cell level. Amazing. Now, another absolutely fundamental antibody-based technique, especially for looking at proteins and complex mixtures, is Western blotting. You mentioned it after immunoprecipitation.
Absolutely. Western blotting, or immunoblotting, is indispensable. Its main purpose is to identify and provide a preliminary quantification of a specific protein within a complex mixture, like a cell lysate.
How does it work, step by step?
First, you take your protein mixture and treat it with a detergent called SDS, which denatures the proteins and gives them a uniform negative charge. Then you separate these denatured proteins based primarily on their molecular weight using gel electrophoresis SDS page. Smaller proteins move faster and farther through the gel.
Okay, proteins separated by size. Then what?
The proteins are then electrophoretically transferred, or blotted, from the fragile gel onto a more robust membrane, usually nitrocellulose or PVDF. This immobilizes the proteins and prevents them from diffusing away.
So now you have an invisible replica of the gel pattern on a membrane. How do you find your specific protein?
This is where the antibody comes in. You incubate the membrane with a primary antibody that specifically recognizes your protein of interest. After washing away unbound antibody, you add a secondary antibody that recognizes the primary antibody and is linked to an enzyme, like horseradish peroxidase, HRP, or a fluorescent tag.
Similar detection as ELISA.
Very similar. If you're using an enzyme, you add a substrate. For HRP, common substrates are chemiluminescent. They produce light when cleaved by the enzyme. This light can be detected by exposing the membrane to photographic film or using sensitive digital imagers. Alternatively, chromogenic substrates produce a colored precipitate directly on the membrane where the antibody is bound. You end up with a band on the membrane corresponding to the size of your target protein.
And the intensity of the band tells you roughly how much protein is there.
Generally, yes. By comparing the band intensity in your sample to controls or standards, you can get a semi-quantitative estimate of the protein's abundance. Western blotting is essential for confirming the presence of a protein, checking its size, which might indicate modifications, and assessing changes in its expression level under different conditions.
Okay, we've covered detecting things with antibodies.
Yeah.
But how do we quantify how strongly an antibody binds to its target? That gets into affinity and kinetics, right? The actual strength of the interaction.
That's correct. Antibody affinity is a quantitative measure of the binding strength between a single antibody binding site and a single epitope on the antigen. It's usually described by the dissociation constant, Kd, typically in molar units. A lower Kd value means higher affinity. The antibody binds more tightly.
How do you measure that?
Well, the classical method, though less common now, is equilibrium dialysis.
Dialysis, like for kidney failure.
Sort of related in principle, using a semi-permeable membrane. For equilibrium dialysis to measure affinity, you have a chamber divided into two compartments by a membrane that allows small molecules, like your antigen if it's small, to pass through, but not large molecules like antibodies.
Okay.
You put your antibody in one compartment and a known concentration of labeled antigen, often radioactive, in the other. The antigen diffuses across the membrane. In the compartment with the antibody, some antigen will bind to the antibody and become trapped because the large antibody-antigen complex can't cross back. Unbound antigen distributes equally between the two compartments.
So the difference in antigen concentration tells you how much is bound.
Exactly. By measuring the concentration of labeled antigen in both compartments at equilibrium, you can calculate the concentration of bound antigen and free antigen. Knowing the antibody concentration, you can then calculate the Kc. It's relatively inexpensive, but can be slow and requires the antigen to be small enough to cross the membrane.
So it tells you the overall affinity when everything settles down. But what if you want to know how fast the antibody binds or how long it stays bound? This is where it gets really scientific, but stick with us, because understanding how an antibody sticks and how long it stays stuck is absolutely critical for things like drug development and diagnostics.
That's precisely the kind of dynamic information provided by the modern standard technique. Surface plasmon resonance, or SPR. Since the mid-1990s, SPR has largely superseded equilibrium dialysis because it's much more rapid, generally more sensitive, and crucially, provides real-time information about the rates of association, binding, and dissociation, unbinding, not just the overall equilibrium affinity. It does require specialized and often expensive instrumentation, though.
Okay, surface plasmon resonance sounds like physics. How does it work conceptually?
It is based on an optical phenomenon. In an SPR instrument, you immobilize one of the binding partners, usually the antigen, onto the surface of a special sensor chip, which has a thin layer of gold. Then you flow the other partner, the antibody, over this surface. The instrument shines polarized light at a specific angle onto the underside of the gold film. At a particular angle, called the resonance angle, the light energy excites electrons in the gold film, creating surface plasmons, and less light is reflected. This resonance angle is extremely sensitive to the refractive index, and therefore the mass, right at the sensor surface.
So when antibodies bind, they add mass.
Exactly. As antibody molecules bind to the immobilized antigen, they increase the mass on the sensor surface, which changes the refractive index and shifts the resonance angle. The instrument detects this shift in real time, and the magnitude of the shift is directly proportional to the amount of antibody bound.
And this gives you the rates.
Yes. The data is typically plotted as a sensorgram, showing the instrument response related to bound antibody versus time. When you first flow the antibody over the surface, the association phase, the signal increases. The slope of this increase tells you about the association rate constant, con, or K1, how fast the antibody binds.
Okay.
When the surface approaches saturation, the curve plateaus. Then, when you switch back to flowing just buffer over the surface, the dissociation phase, the bound antibodies start to come off. The slope of the decreasing signal tells you about the dissociation rate constant, CAR for K2, how fast the antibody releases.
And from those rates, you get the affinity.
Precisely. The equilibrium dissociation constant, Kd, is simply the ratio of the off rate to the on rate. KD equals KUBCon. So SPR gives you not just the overall binding strength affinity, but also the individual kinetic parameters that contribute to it.
That's incredibly powerful. If we connect this to the bigger picture, SPR gives us that dynamic information, not just if an antibody binds, but how fast it binds and releases. Why is that so important, say, for a drug?
It's crucial. For a therapeutic antibody, you might want not only high affinity, low KB, but specifically a very slow off rate. You want the antibody to find its target, bind tightly, and stay bound long enough to exude its biological effect, whether that's blocking a receptor or delivering a toxin. A fast on rate might also be important for quickly neutralizing a target. SPR allows drug developers to select antibodies with the optimal kinetic profile for a specific therapeutic purpose, not just the highest overall affinity.
That makes perfect sense. You need to know the whole binding story. Okay, that's incredibly powerful, moving from a static snapshot to a dynamic movie of molecular interactions.
Yeah.
Now, let's switch gears dramatically and talk about actually seeing cells and their internal structures using various microscopy techniques. And antibodies often play a key role here too, right?
They certainly do. The core idea is simple. We can take antibodies, which provide that exquisite specificity, and conjugate them, link them to molecules that are visible under a microscope. These tags could be enzymes that produce a colored precipitate, fluorescent dyes that glow, or even tiny, dense particles like gold beads for electron microscopy.
Let's start with the enzyme-based methods. What's the difference between immunocytochemistry and immunohistochemistry? They sound very similar.
They are based on the same principle, but differ in the sample type. Both use enzyme-conjugated antibodies to detect specific proteins or antigens within cells or tissues. The enzyme, often horseradish peroxidase, HRP, or alkaline phosphatase, AP, converts a soluble, colorless substrate into an insoluble, colored precipitate right at the site where the antibody is bound.
So you see color where your protein is.
Exactly. Immunohistochemistry, or IHC, is performed on thin sections of intact tissue. This is incredibly valuable because it allows you to see the location of your protein within the context of the tissue's architecture, for example, identifying specific immune cell types infiltrating a tumor or an organ.
Okay, IHCs for tissues. What about ICC?
Immunocytochemistry, or ICC, is essentially the same technique but performed on isolated cells, perhaps cells grown in culture or smeared onto a slide. If you're trying to detect proteins inside the cell, you first need to treat the cells with fixatives and detergents to permeabilize the membranes, allowing the antibodies to get in.
It's like staining a masterpiece. But instead of just painting, you're using these incredibly specific molecular brushes to reveal hidden structures and cell types within the tissue or cell.
That's a beautiful analogy. And you can even do multiplexing, detecting multiple targets at once by using different primary antibodies raised in different species, and then secondary antibodies linked to different enzymes that produce different colored precipitates.
So you could see, say, protein A in brown and protein B in blue in the same tissue section.
Precisely. It allows you to visualize the co-localization or relative distribution of different molecules.
Okay, that's great for light microscopy. But what if you need much higher resolution to see really fine details inside the cell?
For that level of detail, you need to move beyond light microscopy to electron microscopy, or EM. And antibodies can be used here too, in a technique called immunoelectron microscopy. Instead of enzymes or fluorescent dyes, the antibodies are coupled to electron-dense particles, most commonly tiny colloidal gold particles of a specific size.
Gold? Why gold?
Gold atoms are very heavy and scatter electrons strongly, making them show up as distinct dark spots in the electron microscope image, pinpointing where the antibody is bound.
Can you use different sizes?
Yes. That's a key advantage. You can conjugate antibodies specific for different targets to gold particles of different diameters. For example, 5 millimeter versus 15 millimeter. This allows you to visualize the precise subcellular localization of multiple different proteins simultaneously and see their spatial relationships at very high resolution, like seeing two different proteins decorating the same membrane structure.
Incredible resolution, but EM has limitations too, right? Preparing the samples is pretty harsh.
It is. The fixation and embedding procedures required for EM can sometimes damage cellular structures or mask antigenic epitopes, and it's generally more labor-intensive than light microscopy techniques.
Okay, now let's talk about the really vibrant world of immunofluorescence-based imaging. This is where things literally start to glow.
Indeed. Fluorescence microscopy has revolutionized cell biology. The basic principle relies on fluorophores, or fluorescent dyes. These molecules have the property of absorbing light energy at one specific wavelength, the excitation wavelength, and then emitting it back at a longer, lower energy wavelength, the emission wavelength, often in the visible spectrum.
So they absorb blue light and emit green, for example.
Exactly like that. And we have a huge range of tools that leverage this. There are specific dyes that bind directly to certain cellular components, like DAPI or HOCHIST, which bind strongly to DNA and fluoresce blue, allowing us to easily visualize cell nuclei. There are also fluorescently tagged molecules that have natural affinities for specific cellular structures. Phalloidin, for example, binds tightly to filamentous actin, and when conjugated to a fluorescent probe, it lets us visualize the cell's actin cytoskeleton. Annexin A5, which we'll talk about later regarding cell death, binds to a specific lipid on apoptotic cells and can be fluorescently labeled.
And of course, you can attach fluorescent dyes to antibodies.
Absolutely. This is perhaps the most common application in immunofluorescence. Antibodies conjugated to a wide array of different fluorescent dyes emitting green, red, far red, blue light, etc., allow us to specifically label almost any protein of interest for visualization. Streptavidin conjugated to fluorescent dyes is also widely used in conjunction with biotinulated antibodies.
And the real magic happens when you combine these, right? Multiple colors in one image.
That's where it gets truly spectacular. By using multiple antibodies or dyes, each with a distinct fluorescent color, you can simultaneously visualize several different structures or molecules within the same cell or tissue. The resulting multicolor images can reveal complex spatial relationships and dynamic processes. Think of images showing, say, infecting bacteria in red, the host cell's actin cytoskeleton rearranging around them in green, and the cell nucleus in blue, all in one picture.
Amazing. How does the microscope actually handle all those different colors?
A fluorescence microscope uses high-intensity light sources, like mercury lamps or lasers, to provide the excitation light. Then, a sophisticated system of filters and dichroic mirrors comes into play. Filters select the specific wavelength needed to excite a particular dye, and other filters select only the emitted light from that dye to pass through to the detector, or eyepiece, blocking out the excitation light and other wavelengths. Modern microscopes designed for multicolor imaging have complex filter sets and multiple detectors that can simultaneously or sequentially capture the light emitted at several different wavelengths. Software then overlays these separate images, assigning pseudo-colors like red, green, blue to each channel to create the final composite multicolor image.
And it's not just dyes, right? There are fluorescent proteins too.
Yes, the discovery of naturally occurring fluorescent proteins like green fluorescent protein GFP from jellyfish, and subsequent engineering of variants like RFP red, YFP yellow, CFP cyan has been another revelation. Researchers can genetically fuse the gene for a fluorescent protein to the gene of a protein they want to study.
So the cell makes its own fluorescently tagged protein.
Exactly. You can express this fusion protein in cells or even create transgenic animals where, say, all cells of a certain type glow green because they're expressing GFP under the control of a tissue-specific promoter. This allows for tracking protein localization and dynamics in living cells and organisms without needing to add external dyes or antibodies.
Imagine being able to make the inner workings of a cell glow with different colors. That's the magic of fluorescence microscopy. But with all that light bouncing around, especially in thick samples, doesn't it create blurry images? Light coming from above and below where you're trying to focus.
That's a fundamental limitation of standard wide-field fluorescence microscopy, especially when imaging thicker specimens. Light emitted from fluorescent molecules lying outside the focal plane you're interested in still reaches the detector, contributing to out-of-focus haze and blurring the image.
So how do you get sharper images, especially in 3D?
This is precisely the problem solved by confocal fluorescence microscopy. The key innovation in a confocal microscope is a tiny pinhole aperture placed in front of the detector. The optics are designed such that only the light originating from the precise focal plane within the specimen is focused onto this pinhole and can pass through to the detector.
Light from above or below the focal plane gets blocked.
Exactly. Light from out-of-focus planes hits the area around the pinhole and is rejected. This optical sectioning capability results in remarkably clear, sharp images of a single, thin plane within the specimen, free from out-of-focus blur.
And you can build 3D images from these sections.
Yes. Most modern confocal microscopes are laser scanning confocal microscopes. They use lasers for excitation and scan the laser beam across the specimen point by point, collecting the light from each point through the pinhole. By scanning across a plane, XY scanning, and then moving the focal plane up or down, Z scanning, the microscope can collect a stack of these optical sections. Powerful computer software then reconstructs these sections into a high-resolution, three-dimensional image of the sample.
That's amazing clarity. But I've heard of something potentially even better for thick tissues or live imaging, multiphoton fluorescence microscopy. What's the advantage there? How is it different?
Multiphoton microscopy, sometimes called two-photon microscopy, is a variation on confocal that offers even greater 3D resolution and several key advantages, especially for imaging deep within living tissues. It addresses a remaining issue with standard confocal. The excitation laser beam still excites fluorophores all along its path through the specimen, even outside the focal plane. This can lead to photobleaching, damage to the fluorescent molecules, and phototoxicity, damage to the cells, in those out-of-focus regions.
Okay, so how does multiphoton avoid that?
It uses a fundamentally different excitation principle. Instead of using a single high-energy photon, e.g. blue or UV light, to excite the fluorophore, it uses long-wavelength, lower-energy lasers, typically in the infrared range. The key is that exciting the fluorophore requires the simultaneous absorption of two or more of these low-energy photons.
Two photons hitting at the exact same time, isn't that unlikely?
It is extremely unlikely under normal laser intensities. It only becomes probable at the very precise point where the laser beam is most tightly focused, the focal point. This means that fluorescence excitation occurs only within the tiny volume of the focal spot and essentially nowhere else along the laser path.
So no out-of-focus excitation.
Exactly. This has several major benefits. First, reduced photobleaching and phototoxicity, because excitation is confined to the focal plane, allowing for longer imaging times in living samples. Second, because excitation is inherently localized to the focus, you get excellent optical sectioning without needing a pinhole, which actually improves light collection efficiency. And third, and perhaps most importantly for immunologists wanting to look inside tissues, the longer wavelength infrared light scatters much less than visible or UV light as it passes through tissue. This allows multiphoton microscopy to penetrate much deeper into thick specimens hundreds of microns deep, compared to maybe tens of microns for standard confocal.
Deeper, gentler, and sharper. That sounds ideal for looking inside living tissues. Which brings us to intravital imaging. This is literally watching the immune system in action inside a living, breathing organism, right? Using these multiphoton microscopes.
This is where dynamic imaging truly comes alive. Intravital imaging allows researchers to maintain organs, like lymph nodes or sections of the intestine, with their blood and lymphatic circulation intact, usually on a specialized, warmed microscope stage. Using a multiphoton microscope, they can then generate 3D time-lapse images of fluorescently labeled cells moving and interacting within these tissues. It's like watching a movie of the immune system unfolding in real time, in its native environment.
What kind of things can you see?
You can directly observe immune cell trafficking, how lymphocytes enter and leave lymph nodes, how they migrate within tissues, how T cells search for antigen-presenting cells, the dynamics of T-cell APC interactions, the formation of immune synapses, the killing of target cells by cytotoxic lymphocytes, neutrophils swarming to sites of infection. The possibilities are incredible. But, like any powerful technique, the data needs critical interpretation. When you see these amazing movies, you have to ask, okay, what cells are actually labeled here? What fraction of the total population am I seeing? How stable is the fluorescent label over time? How and when was the antigen or stimulus introduced? Is it a physiological model? What specific tissue or microenvironment am I looking at? Asking these critical questions is essential for drawing accurate conclusions from this visually compelling data.
Now, context is everything. What have been some key discoveries from intravital imaging?
It's provided visually stunning confirmation, or sometimes refutation, of hypotheses generated from more static or in vitro studies. It's revealed unexpected locations for initial immune interactions, like the subcapsular sinus of lymph nodes. It's shown surprising cell types participating in certain responses, like neutrophils playing early roles. It's mapped out intricate cell motility patterns, for example, within germinal centers, where B cells undergo affinity maturation. It allows us to directly address fundamental questions about how immune responses initiate, progress, and resolve within the complex 3D architecture of tissues.
It sounds incredibly powerful, despite any limitations. Even just as an educational tool, seeing a video of T cells swarming must clarify things in a way that diagrams just can't.
Absolutely. A short intravital imaging video can convey the dynamism and complexity of the immune system in minutes. It really brings the whole process to life. But it's good to remember the Avedon quote, "All photos are accurate, none of them is the truth." Moving images get us closer, but there's still a representation of a manipulated experimental system.
A great point. Now, staying within the realm of visualization, but shifting focus from proteins in cells to the blueprint itself, DNA. How do we visualize DNA sequences and their organization within the nucleus in their native chromatin structure?
That's another crucial area, as the 3D organization of DNA profoundly impacts gene expression and rearrangement, especially important in lymphocyte development. One technique is 3D fluorescence in situ hybridization, or 3D FESH.
FESH, like fishing for genes.
Kind of. In FESH, you use fluorescently labeled probes, usually short RNA or DNA sequences that are complementary to specific DNA sequences in the genome. These probes hybridize or bind to their target sequences within intact cell nuclei.
So you can light up specific genes.
Exactly. And in 3D FESH, you use sophisticated microscopy, often confocal, to visualize the 3D positions of these fluorescent spots within the nucleus. You can use probes of different colors for different genes. For instance, immunologists have used this to study the relative positions of the variable V, diversity D, and joining J gene segments that make up B cell receptors, antibodies, or T cell receptors.
What did they find?
They could observe, for example, how large loops of the immunoglobulin chromosome physically contract during B cell development, bringing distant V gene families closer to the D and J segments, presumably to facilitate the VDJ recombination process that creates antibody diversity. It provides a direct visual readout of large-scale chromatin rearrangements.
So we can literally see the DNA folding and rearranging on a large scale. That's remarkable. What about zooming in? How do you figure out which specific proteins are bound to which specific DNA sequences at any given time? That seems critical for gene regulation.
Absolutely critical. The main technique for that is chromatin immunoprecipitation, or CHEAT-IP.
Okay, CHEAT-IP, how does that work?
In CHEAT-IP, you first treat living cells with a chemical, usually formaldehyde, that cross-links proteins to nearby DNA, essentially freezing protein-DNA interactions as they exist in the cell. Then you isolate the chromatin and shear it into small fragments.
Break the DNA up.
Right. Next, you use an antibody that specifically recognizes the DNA-binding protein you're interested in, maybe a transcription factor or a modified histone. This antibody is used to immunoprecipitate that protein. Because the protein is cross-linked to the DNA it was sitting on, you pull down the protein and its associated DNA fragments.
Ah, IP again, but this time pulling down protein DNA complexes.
Exactly. You then reverse the cross-links to release the DNA, purify it, and identify the DNA sequences. Originally, this was done by PCR for specific candidate sequences, but now it's almost always coupled with high-throughput sequencing called ChIP-seq. This gives you a genome-wide map of all the locations where your protein of interest was bound.
Wow, a map of protein binding sites across the whole genome.
It's incredibly powerful for understanding gene regulatory networks.
And what about even longer range interactions? How does DNA looping bring distant regulatory elements close to gene promoters? Can we map those interactions too?
Yes, that's where chromosome conformation capture 3C technologies come in. There are several variations, but a widely used genome-wide version is called Hi-C.
I see. What's the goal here?
The goal of Hi-C is to create a comprehensive map of all the physical interactions between different DNA segments within the nucleus, revealing the 3D architecture of the genome. It tells you which DNA regions, even if they are millions of base pairs apart on the linear chromosome sequence, are actually spatially close to each other inside the nucleus due to folding and looping.
How does it capture that proximity?
It also starts with cross-linking, fixing those spatial interactions. Then, the DNA is cut with a restriction enzyme. Critically, the sticky ends created are then filled in with biotin-labeled nucleotides. This marks the ends of the original fragments. Then, under very dilute conditions that favor ligation between cross-linked fragments, the DNA ends are ligated back together.
So DNA bits that were physically close get stitched together into a new hybrid molecule.
Precisely. You then shear the DNA, specifically purify the fragments containing biotin, which represent those original ligation junctions, and sequence them using high-throughput sequencing. By mapping the pairs of sequences that got joined together, you can build up a genome-wide interaction matrix, showing how frequently any two regions of the genome interact. This reveals domains, loops, and compartments that define the 3D organization of chromosomes.
That sounds incredibly complex, but yields a fascinating picture of genome architecture. Okay, let's zoom out again from molecules and structures to analyzing whole cells, particularly in large populations. When you want to analyze and sort hundreds of thousands, even millions of cells based on the characteristics, you turn to flow cytometry and fluorescence-activated cell sorting, or FACS. This is really the bread and butter for experimental immunologists, isn't it?
It absolutely is. You could argue that flow cytometry is the sine qua non, the indispensable tool of the modern immunologist. Developed largely by Leonard and Leonore Herzenberg and colleagues starting in the late 60s and becoming widespread in the early 80s, it found some of its earliest and most critical applications in analyzing lymphocyte subpopulations, like the ratio of CD4 plus helper T cells to CD8 plus cytotoxic T cells in patients with the then emerging HIV/AIDS epidemic.
So what's the core function? What does it measure?
Its core function is to rapidly analyze multiple physical and fluorescent characteristics of single cells as they flow in a fluid stream past one or more laser beams. It can analyze hundreds of thousands, even millions of cells in minutes. The two main types of measurements are light scatter and fluorescence.
Okay, let's start with light scatter. What does that tell you about a cell?
As each cell passes through the laser beam, it scatters the light. Detectors measure the scattered light in two main directions. Forward scatter, or FSC, is the light scattered in the same direction as the laser beam. The amount of FSC is roughly proportional to the cell size. Larger cells scatter more light forward.
Bigger signal, bigger cell. What about side scatter?
Side scatter, or SSC, is the light scattered at an angle, typically 90 degrees, to the laser beam. SSC is more related to the cell's internal complexity or granularity. Cells with lots of internal membranes or granules, like neutrophils or eosinophils, will have higher SSC than cells with a simpler internal structure, like lymphocytes.
So just from scatter alone, FSC versus SSC, you can often distinguish major cell populations, like lymphocytes from granulocytes.
Absolutely. A standard FSC versus SSC plot is often the first step in analyzing flow cytometry data, allowing you to visually separate different cell types based on these physical properties before even looking at fluorescence. The instrument also measures the characteristics of the electronic pulse generated by each scatter or fluorescence signal, its height, width, and area, which can provide additional information, for instance, helping to distinguish single cells from doublets or clumps.
Okay, that's the physical part. And the fluorescence part is where the antibodies come back in tagged with those fluorescent dyes we talked about.
Exactly. If the cells have been stained with fluorescently labeled antibodies that bind to specific cell surface markers or intracellular proteins after permeabilization, the laser light will excite these fluorophores. The emitted fluorescent light is then collected, filtered, and measured by sensitive detectors, photomultiplier tubes, or PMTs.
How does it keep the cells single file? They must be moving fast.
That's achieved through a process called hydrodynamic focusing. The sample stream containing the cells is injected into a faster-flowing outer stream of sheath fluid, usually saline. This narrows the sample stream dramatically, forcing the cells to align and pass through the laser interrogation point one by one, ensuring accurate single-cell measurements.
And how does it sort out all the different colors of fluorescence if you're using multiple antibodies?
That's where the complex optics come in the beam splitters, mirrors, and filters we discussed earlier in the context of microscopy. After the cell passes the laser, the scattered light, FSC, SSC, and the emitted fluorescent light are collected. Dichroic mirrors split the light path based on wavelength, directing different color ranges towards specific detectors. Bandpass filters in front of each detector further refine the wavelengths measured, ensuring that, for example, green fluorescence from FITC is measured by one detector, red fluorescence from PE by another, and so on.
A sophisticated light sorting system inside the machine. And all this data scatter and multiple fluorescence channels for every single cell gets stored.
Yes, that's crucial. The instrument records all the measured parameters for each individual cell as it passes through. This raw data is stored in a standardized format called list mode data, usually FCS files. This is incredibly powerful because it allows researchers to go back to the data later and reanalyze it in numerous ways using specialized software without having to rerun the experiment.
How do you typically analyze this data? Plotting dots.
Dot plots are a primary tool. After maybe using the FSC-SSC plot to initially identify a broad population of interest, like lymphocytes, using a process called gating, you can then create plots showing two different fluorescence parameters against each other. For example, plotting CD4 fluorescence versus CD8 fluorescence allows you to easily identify and quantify CD4 plus T cells, CD8 plus T cells, double positive cells, if any, and double negative cells within your lymphocyte gate.
And modern machines can measure many more parameters than just 2 or 3 colors.
Oh yes. State-of-the-art flow cytometers use multiple lasers emitting at different wavelengths and an array of detectors capable of measuring 20, sometimes even 40 or more, different fluorescence parameters simultaneously, in addition to scatter. Analyzing this high-dimensional data requires sophisticated software tools like FlowJo or specialized algorithms like SPADE or UMAP-TSE, which help visualize complex populations and relationships in ways simple 2D plots cannot.
But you mentioned earlier the challenge of spectral overlap when using many fluorescent dyes. How does that limit things?
That remains a significant challenge. Because fluorescent dyes emit light over a range of wavelengths, not just a single sharp peak, the emission spectrum of one dye, say FITC, primarily green, might spill over into the detector intended for another dye, say PE, primarily yellow-orange. This spectral overlap makes it harder to accurately distinguish the signals from different fluorophores, especially as you add more colors.
Can you correct for it?
Yes, instruments use a process called compensation, where software electronically subtracts the known spillover from adjacent channels. However, compensation becomes increasingly complex and less accurate as the number of colors increases, and significant overlap ultimately limits the practical number of distinct parameters you can reliably resolve simultaneously, often cited as being around 1820 proteins per cell with conventional fluorescence flow cytometry. This limitation is a major driver for developing alternative technologies.
It's amazing how flow cytometry and monoclonal antibody technology really grew up together and fueled each other's development.
They absolutely did. The ability to produce highly specific monoclonal antibodies against cell surface markers provided the perfect reagents for flow cytometry, and flow cytometry provided the ideal platform for utilizing these antibodies to dissect the complexity of the immune system at the single cell level. It's a cornerstone for both research and clinical diagnostics used for classifying leukemias, monitoring HIV progression by counting CD4 cells, analyzing stem cells for transplantation, and diagnosing various blood disorders.
This is where we go from observing a few cells to quantifying hundreds of thousands or even millions in minutes. It's the ultimate numbers game in cell biology. But if fluorescence has limits due to spectral overlap, what's the next step for measuring even more parameters per cell?
That brings us to CytoF, which stands for cytometry by time of flight. It's a technology that brilliantly combines the antibody-based cell labeling strategy of flow cytometry with the high resolution of mass spectrometry.
Mass spectrometry instead of fluorescence. How did that work?
Instead of tagging antibodies with fluorescent dyes in CytoF, antibodies are conjugated with highly purified, stable isotopes of rare earth metals. These metals, like lanthanum, ytterbium, gold, etc., are not naturally abundant in biological systems and have unique atomic masses.
So each antibody carries a unique atomic weight tag.
Exactly. You stain your cells with this panel of metal-tagged antibodies. The cells are then introduced one by one into the CytoF instrument, where they are nebulized into tiny droplets and pass through an incredibly hot, inductively coupled plasma, ICP, around 7,000 Kelvin.
That vaporizes the cell.
Completely. It obliterates the cell, breaks all molecular bonds, and ionizes the atoms, creating a cloud of ions derived from each individual cell. This ion cloud, containing the metal isotope reporters, is then sent into a time-of-flight mass spectrometer.
Time of flight. Heavier things travel slower.
Precisely. The ions are accelerated by an electric field and then drift through a flight tube to a detector. Ions with different mass-to-charge ratios will travel at different speeds. Lighter ions arrive faster, heavier ions arrive slower. The instrument measures the arrival time for each distinct metal isotope, and the intensity of the signal for each isotope tells you how many antibodies carrying that metal tag were bound to the original cell.
And because the mass peaks for these isotopes are incredibly sharp and distinct, you don't have the overlap problem like fluorescence.
Exactly. The mass peaks are very narrow, allowing for the simultaneous detection of many more parameters than fluorescence flow cytometry. Current CytoS systems can routinely analyze at least 45 simultaneous parameters, and potentially up to 100, providing an unprecedented depth of information from each single cell.
That's a huge leap in dimensionality. But you mentioned trade-offs earlier. As we demand more information from each single cell, what are the trade-offs with CyTOF?
The main trade-offs are speed and the type of data collected. CyTO instruments are currently significantly slower than high-end fluorescence flow cytometers. While a fast FACS machine might analyze 50,000 cells per second or more, CyTOF typically maxes out around 1,000 cells per second. This makes it less suitable for analyzing extremely rare cell populations.
Okay, slower processing. What else?
CyTOF also, by its nature, destroys the cell, so you can't sort living cells for downstream functional studies like you can with FACS. And importantly, current CytoS technology doesn't provide the light scattering information, FSC and SSC, that's so useful in conventional flow cytometry for basic cell identification based on size and granularity. You lose that physical context. So it's a trade-off. Incredible parameter depth versus speed, cell viability, and physical scatter data.
It's about choosing the right tool for the job, weighing those pros and cons. Sometimes you need the depth, sometimes the speed or the scatter. Okay, so FACS and Cyto-F analyze cells one by one. What if you just need to separate a large population of cells based on one or two markers, maybe more gently?
That's where magnetic activated cell sorting, or MANCS, often comes in. It's another way to separate cells based on antibody binding, but using magnetic properties instead of fluorescence and high-speed sorting.
Magnetic beads again, like in immunoprecipitation.
Yes, very similar. Antibodies specific for a cell's surface marker are conjugated to tiny paramagnetic beads, only magnetic in the presence of a magnetic field. You incubate your mixed cell population with these antibody-coated magnetic beads.
So the cells you want get magnetically tagged.
Right. Then you pass the entire cell suspension through a column that's placed within a strong magnetic field. The column matrix, often a ferromagnetic wool mesh, creates a high magnetic gradient. The magnetically labeled cells are retained in the column stuck to the mesh, while the unlabeled cells simply flow through.
So you collect the flow through for the negative population.
Exactly. Then you remove the column from the magnetic field. Without the magnet, the beads are no longer magnetic, and the retained cells can be easily flushed out of the column with buffer, giving you your positively selected population.
That sounds much gentler than shooting cells through a nozzle at high speed like FACS.
It is generally considered extremely gentle, which can be important for sensitive cells. It's also very good for processing large numbers of cells relatively quickly in bulk, a batch separation method.
How does it compare to FACS in terms of purity or accuracy?
FACS is generally more precise and achieves higher purity because it interrogates and sorts cells one at a time, making fewer mistakes. MACS, being a bulk method, might trap some non-target cells or let some target cells flow through. However, MACS is faster for processing very large numbers and, as mentioned, gentler.
Can you combine them?
Very often, yes. Immunologists frequently perform an initial enrichment using MACS to isolate a large population of interest, and then follow up with FACS to achieve very high purity sorting of specific subpopulations within that enriched fraction. It's a common strategy, for example, in preparing highly purified stem cells for transplantation.
Makes sense. You use the net first, then the scalpel. Okay, we've covered analyzing and sorting cells based on their surface markers. Now let's shift focus to understanding what the cells are actually doing internally specifically. Fundamental processes like cell division, proliferation, and cell death, apoptosis. How do we measure those?
These are absolutely critical measurements in immunology. Lymphocyte proliferation is a hallmark of an adaptive immune response, and controlled cell death is essential for eliminating infected cells and contracting the immune response after the threat is gone. Immunologists have developed many assays for both.
Let's start with proliferation. How did they first measure cell division?
The classic first routine method was tritiated thymidine uptake. Thymidine is one of the building blocks of DNA. Tritiated thymidine is simply thymidine where some hydrogen atoms are replaced with tritium, a radioactive isotope of hydrogen.
So feed dividing cells radioactive DNA building blocks.
Essentially, yes. Dividing cells are actively synthesizing DNA, so if you add 3,8-thymidine to the culture medium, they will incorporate it into their newly made DNA. After a period of incubation, you release the cells, precipitate the DNA onto filters, wash away any unincorporated 3,8-thymidine, and then measure the amount of radioactivity trapped on the filters using a scintillation counter. The amount of radioactivity is proportional to the amount of DNA synthesis, and thus cell division.
Effective, but again, radioactivity concerns.
Precisely. Safety, waste disposal, and regulatory hurdles associated with radioactivity led to the development of non-radioactive alternatives. One popular category is colorimetric assays, like the MTT assay.
How does MTT work? No radioactivity here.
Right. MTT is a yellow, water-soluble tetrazolium salt. Living, metabolically active cells possess mitochondrial oxidorreductase enzymes that can cleave the tetrazolium ring of MTT, converting it into an insoluble purple compound called formazan. These purple crystals accumulate within the cell.
So more live cells, more purple.
Exactly. You then add a detergent to solubilize these purple formazan crystals, and you can measure the intensity of the purple color using a standard spectrophotometer, measuring absorbance at around 570 millimeter. The amount of purple color is directly proportional to the number of metabolically active cells in the culture. So you can track proliferation by seeing an increase in color over time, or conversely, measure cell death or toxicity by seeing a decrease in color compared to untreated controls.
Clever use of cell metabolism. What about other DNA precursors? I've heard of BRDU.
Yes, bromodeoxyuridine, or BRDU, is another very important tool. BRDU is an analog of thymidine. It looks very similar structurally. Like thymidine, BRDU gets incorporated into newly synthesized DNA during the S phase DNA replication of the cell cycle.
So cells that divided will have BRDU in their DNA.
Correct. The key difference is that you can then use a specific antibody that recognizes BRDU. So after exposing cells to BRDU, you can fix and permeabilize them, stain with an anti-BRDU antibody, often fluorescently labeled, and then use flow cytometry or microscopy to identify and quantify the cells that incorporated BRDU, i.e. the cells that were actively dividing.
So it labels the dividing cells for later detection. Does BRDU do anything else?
Interestingly, yes. Incorporation of BRDU into DNA makes the cells more sensitive to light-induced damage, a phenomenon called photolysis. This property has been used experimentally to selectively kill off newly dividing cells in a population. Newer analogs like EDU, 5-ethanol-2-deoxyuridine, have also been developed, which can be detected using click chemistry, sometimes offering advantages in sensitivity or ease of use over BRDU.
Okay, so thymidine uptake, MTT, BRDU. These tell you if cells are dividing or have divided. What about figuring out where a cell is in the cell cycle right now? G1, S, G2, M?
For determining cell cycle status, the most common method uses DNA-binding fluorescent dyes like propydium iodide, or PI. PI is a flat, planar molecule that intercalates or slips into the DNA double helix. Crucially, it binds quantitatively. The amount of PI fluorescence emitted by a cell is directly proportional to the total amount of DNA it contains.
How do you measure that with flow cytometry?
You stain fixed and permeabilized cells with PI and run them on a flow cytometer, detecting the red fluorescence, usually in the FL2 or FL3 channel. Importantly, for cell cycle analysis, you typically analyze the area of the fluorescence pulse signal, not just its height, as this gives a better measure of total DNA content.
And different phases have different DNA amounts.
Exactly. Yeah. Cells in the G0 or G1 phase, resting or before DNA replication, have a normal diploid DNA content, 2N. Cells in the S phase are actively replicating their DNA, so they will have an intermediate amount of DNA between 2N and 4N. Cells in G2, after replication but before division, or M phase, mitosis, will have doubled their DNA content 4N.
What about dying cells?
Apoptotic cells often fragment their DNA, so they typically show up as a population with less than 2N DNA content, often called the sub-G1 peak. So a histogram plotting the number of cells versus PI fluorescence intensity will show distinct peaks corresponding to GOG1, S, and G2M phases, plus potentially a sub-G1 peak.
That gives you a snapshot of the whole population's proliferative state. Are there other dyes like PI?
Yes, other DNA-binding dyes like DAPI, Hoch33342, or 7AD can be used similarly for cell cycle analysis, sometimes offering advantages like being usable in live, unfixed cells, Hochist, or having different spectral properties. A key thing in cell cycle analysis is careful gating to exclude cell doublets or clumps, which can be mistakenly interpreted as G2M cells because they have double the DNA signal. So this raises an important question. How does knowing a cell's DNA content or replication status tell us about its overall health and response to a stimulus?
Right. If you stimulate T cells, you'd expect to see them move from G0, G1 into S and G2M, right?
Yeah, exactly. An accumulation of cells in S and G2M indicates active proliferation in response to the stimulus. Conversely, if a treatment causes cell cycle arrest, you might see cells pile up in G1 or G2. And as mentioned, an increase in the sub-G1 population suggests cell death. So cell cycle analysis provides dynamic readout of cellular responses.
Now, there's one more proliferation assay that seems particularly elegant, CFSE. It lets you track actual cell divisions, like counting generations.
CFSE, or carboxyfluorescein secanometyl ester, is indeed a very powerful and widely used dye for tracking cell divisions. It's a cell permit dye that, once inside the cell, gets modified by intracellular enzymes, esteroses, that cleave off acetyl groups, trapping the fluorescent CFSE molecule inside. It then covalently attaches to intracellular proteins.
So it stains the whole cell's insides green.
Pretty much, yes. And it does so brightly and without harming the cell. The real magic of CFSE lies in what happens when the cell divides. The total fluorescence is distributed roughly equally between the two daughter cells.
Meaning each daughter cell is half as bright as the parent.
Exactly. With each successive cell division, the amount of CFSE fluorescence per cell is precisely halved.
Wow. So if you analyze CFSE-labeled cells by flow cytometry after a few days of stimulation,
You'll see a series of distinct peaks on a fluorescence histogram. The brightest peak, often furthest to the right, represents the cells that haven't divided at all, generation 0. The next peak to the left, with exactly half the fluorescence intensity, represents cells that have divided once, generation 1. The next peak is generation 2, then generation 3, and so on.
This is truly ingenious. Imagine painting a cell and then watching how that paint gets diluted as it divides, giving you a precise count of its generations.
It provides incredibly detailed information about the proliferative history of the cell population, not just if they divided, but how many times. And combined with FECS, you can even physically sort cells based on the number of divisions they've undergone and then perform further analyses, like measuring gene expression in cells that have divided exactly 3 times versus 5 times.
That's amazing resolution. Okay, so that covers proliferation pretty well. Now let's flip the coin and talk about measuring cell death. As you said, it's just as important in immunology.
Absolutely. Regulated cell death, primarily apoptosis, is crucial for normal immune function development and homeostasis. And many immune effector mechanisms, like those mediated by cytotoxic T lymphocytes, CTLs, or NK cells, work by inducing death in target cells. So we need reliable ways to measure it.
What was the classic method for measuring cell killing, say, by CTLs?
For decades, the undisputed gold standard was the chromium-51 release assay. It was the method used in the Nobel Prize-winning experiments by Doherty and Zinkernagel that first demonstrated MHC restriction of T-cell killing.
Chromium-51, another radioactive isotope.
Yes. In this assay, the target cells, the cells you expect to be killed, are preloaded with radioactive sodium chromate, Na251CrO4, which they take up into their cytoplasm. After washing away excess chromium, these labeled target cells are mixed with the effector cells, like CTLs.
And if the CTLs kill the target cells?
The target cell membrane becomes leaky, and the radioactive 51CR trapped inside gets released into the culture supernatant. You then collect the supernatant and measure the amount of radioactivity released using a gamma counter. By comparing the release from cells incubated with effectors to the spontaneous release, targets alone, and maximum release, target sliced with detergent, you can calculate the percentage of specific killing.
Still used today? Or are there non-radioactive options?
It's less common now due to the radioactivity, but the principle is sound. A popular non-radioactive alternative involves labeling the target cells with CFSE, as we just discussed, or another stable cytoplasmic dye. When the cells are killed, the dye leaks out, and you can measure the amount of fluorescence released into the supernatant using a fluorescence plate reader. These assays measure all forms of cell death that lead to membrane rupture, necrosis, late apoptosis.
Okay, so those measure general cell lysis. What if you were to specifically measure apoptosis, that programmed form of cell death?
There are several assays specifically designed to detect hallmarks of apoptosis. One very common one uses fluorescently labeled annexin A5.
Annexin A5. What did that bind to?
During the early stages of apoptosis, a specific membrane phospholipid called phosphatidylserine, PS, which is normally confined to the inner leaflet of the plasma membrane, gets actively flipped to the outer leaflet, exposing it on the cell surface. Annexin A5 is a protein that has a high affinity for binding to PS, but only in the presence of calcium.
So if PS is on the outside, annexin A5 will stick.
Exactly. By using annexin A5 that's been conjugated to a fluorescent dye, like FITC or APC, you can label cells that are undergoing apoptosis. These cells can then be detected by immunofluorescence microscopy, or more commonly quantified by flow cytometry. Often, annexin A5 staining is combined with a viability dye like PI or 7AD.
Why combine them?
Because cells in late apoptosis or necrosis will lose membrane integrity and also become permeable to PI. So cells that are annexin V positive but PI negative are considered early apoptotic. Cells positive for both are late apoptotic or necrotic. Cells negative for both are live and healthy. This combination gives you a more nuanced picture of cell death stages.
That makes sense. What's another way to detect apoptosis? Something about DNA fragmentation.
Yes, another key biochemical hallmark of apoptosis is the progressive degradation of chromosomal DNA by endogenous endonucleases. This generates numerous DNA strand breaks, creating many free 3-fat hydroxyl ends within the nucleus. The TNL assay detects this DNA fragmentation.
TNL, what does that stand for?
It stands for terminal deoxynucleotidyl transferase DUTP-NIC-N labeling. It's a bit of a mouthful. It uses an enzyme called terminal deoxynucleotidyl transferase, TDT. TDT has the unusual property of being able to add nucleotides onto the 3-foot end of DNA in a non-template dependent manner.
So it just sticks nucleotides onto broken DNA ends.
Precisely. In the TNL assay, you provide TDT along with labeled DUTP nucleotides, often DUTP linked to biotin or a fluorescent dye. TDT incorporates these labeled DUTPs onto the numerous DNA breaks present in apoptotic cells. The amount of incorporated label, which can be detected by fluorescence microscopy or flow cytometry, is therefore a measure of the extent of DNA fragmentation and thus apoptosis.
So expose PS with annexin V or breaking DNA with tunal. Any other ways to measure apoptosis? Maybe by looking at the executioner enzymes themselves.
Yes, that's the third major approach, measuring the activity of casposes. Casposes are a family of cysteine proteases that act as the central executioners of the apoptotic program. Different casposes are activated in a cascade depending on the apoptotic stimulus. For example, caspase 8 is activated early in the death receptor pathway, while caspase 9 is activated by the mitochondrial pathway, both converging on effector casposes like caspase 3.
Can you measure their activity directly?
Yes. There are commercially available tests that contain synthetic caspase substrates linked to a fluorescent or colorimetric reporter molecule. When an active caspase cleaves the substrate, the reporter is released, generating a measurable signal. Alternatively, you can use specific antibodies that recognize only the cleaved active forms of specific caspases, often detected by Western blotting or flow cytometry. Measuring caspase activation provides a direct readout of the apoptotic machinery being engaged.
So, multiple ways to measure cell death, each looking at a different aspect of the process. That gives researchers options depending on the question. Okay, we've explored antibodies, assays, visualization, cell analysis. Let's move to the ultimate level of complexity and control. Genetic manipulation and applying these tools in whole animal systems. And we have to start with the technology that's completely changed the game, CRISPR-Cas9.
Absolutely. CRISPR-Cas9 truly sits at the intersection of in vitro and in vivo methodologies because it's been used so effectively in both cell culture and living organisms. Few technological innovations in recent memory have had a greater, more rapid, or more wide-ranging impact on biological research.
And incredibly, it originated as a bacterial immune system, right?
Wow.
Fighting off viruses.
That's exactly right. The CRISPR, clustered regularly interspaced short palindromic repeats system, evolved in bacteria and archaea as a form of adaptive immunity against invading bacteriophages, viruses that infect bacteria, and plasmids.
How does it provide immunity?
When a bacterium survives an encounter with a new phage, it often integrates a short segment of the phage's DNA, called a spacer, into the CRISPR locus in its own chromosome. This locus contains an array of these captured spacer sequences, interspersed with identical repeat sequences.
Building a library of past invaders.
Kind of like a genetic most wanted list. When the bacterium encounters that same phage again, the CRISPR locus is transcribed into RNA, and this RNA is processed into smaller guiding RNAs, called CRISPR RNAs, cRNAs, each containing one of the spacer sequences.
Okay, guide RNAs. Then what?
These eRNAs then associate with CRISPR-associated, or Cas, proteins. These Cas proteins are enzymes, often nucleuses that can cut DNA. The RNA guides the Cas protein complex to the corresponding sequence in the invading phage DNA.
Finds the matching sequence on the most wanted list.
Exactly. Once the complex binds to the target DNA sequence in the phage genome, the Cas nucleus, like the famous Cas9, makes a double-strand break in the phage DNA, effectively destroying it and neutralizing the infection. The power for genetic engineering comes from realizing that we can program the system with any guide RNA sequence to target any desired DNA sequence for cutting.
So repurposing a bacterial defense system for precision genome editing. What are the core components scientists typically use in the lab, specifically for the type 2 CRISPR-Cas9 system?
The workhorse type 2 CRISPR-Cas9 system, derived from Streptococcus pyogenes, has two main components that need to be delivered into the target cell. One, the Cas9 protein itself, which is the enzyme that actually cuts the DNA. Two, a guide RNA, gRNA. In the engineered lab system, this is often a synthetic single guide RNA, sgRNA, that combines the functions of the natural cRNA, which contains the 20 nucleotide sequence that matches the target DNA, and another RNA called the tracer RNA, which helps scaffold the Cas9 protein.
So Cas9 does the cutting, and the guide RNA tells it exactly where to cut.
That's the essence of it.
But wait, if the bacteria incorporate bits of phage DNA into their own CRISPR locus, how does the CRISPR-Cas9 system avoid constantly cutting the bacterium's own DNA at that locus? That seems like a major self-destruct problem.
An excellent question, and the system has an elegant solution. The PAM sequence. For the Cas9 nucleus to efficiently bind and cut a target DNA sequence, it requires not only the correct match with the guide RNA spacer sequence, but also the presence of a short, specific DNA sequence motif immediately downstream of the target site. This is called the protospacer adjacent motif, or PAM.
Okay, a PAM site is needed next to the target.
Right. For the commonly used S. pyogenes Cas9, the PAM sequence is typically 5' in G, where N is any nucleotide. The crucial part of the self-protection mechanism is that during the process where the bacterium acquires new spacers from foreign DNA, it never incorporates the associated PAM sequence into its own CRISPR locus. Therefore, the CRISPR locus itself lacks the PAM site required for Cas9 cutting, protecting the host genome from self-destruction.
Incredibly clever. The system needs two keys to turn, the guide sequence match and the PAM nearby. Okay, so how do scientists harness this targeted cutting ability in the lab? What can you do with CRISPR-Cas9?
The simplest and most common application is gene inactivation, or creating a gene knockout. You introduce Cas9 and a guide RNA targeting your gene of interest into cells. Cas9 cuts the DNA at the target site, creating a double-strand break.
Then the cell tries to fix it.
Yes, the cell's own DNA repair machinery kicks in. The most common repair pathway for such breaks is called non-homologous end joining, or NHEJ. NHEJ is fast, but it's notoriously error-prone. It often introduces small insertions or deletions, called indels, at the repair site.
And those errors mess up the gene.
Often, yes. If the indel causes a frame shift in the protein coding sequence, it usually leads to a premature stop codon and results in a non-functional protein product, effectively knocking out the gene. This is widely used in genome-wide screens to systematically investigate the function of thousands of genes by knocking them out one by one.
Okay, so you can break genes easily. What if you want to make a precise change, like correcting a mutation or inserting a new sequence?
You can do that too by hijacking a different DNA repair pathway called homology-directed repair, or HDR. HDR uses a template to accurately repair double-strand breaks. So, along with Cas9 and the guide RNA, you also introduce a donor DNA template into the cell. This donor template contains the desired genetic change, or the corrected sequence, or a sequence you want to insert, flanked by sequences that are homologous to the DNA on either side of the Cas9 cut site.
So the cell uses your template to fix the break.
Exactly. The cell's HDR machinery uses the donor DNA as a template to repair the break, precisely incorporating your desired change into the genome at the target location. This is much less efficient than NHEJ, but it allows for precise gene editing correcting mutations, inserting tags like GFP onto proteins, or even introducing specific disease-causing mutations to model human diseases.
Incredible precision. Could you target multiple genes at once?
Yes, that's called multiplexing. You can introduce Cas9 along with several different guide RNAs targeting different genes simultaneously, allowing for multiple genetic changes in the same cell or organism.
And what about using CRISPR without actually cutting the DNA? You mentioned dead Cas9.
Right, that's another powerful extension. By engineering mutations in Cas9 that inactivate its DNA cutting domains, you create a dead Cas9, or dCas9. This dCas9 protein can still be targeted to specific DNA sequences by a guide RNA, but it doesn't cut the DNA.
So it just sits there. What's the use of that?
You can fuse other functional domains to dCas9. For example, you could fuse a transcriptional repressor domain to dCas9 to specifically silence a target gene, CRISPR interference or CRISPR, or fuse a transcriptional activator to turn a gene on, CRISPR activation or CRISPR. You could fuse dCas9 to fluorescent proteins, like GFP, to visualize the location of specific DNA sequences in the nucleus in living cells. Or fuse it to enzymes that modify chromatin, epigenetic editing, to study how those modifications affect gene expression, all without altering the underlying DNA sequence. It's incredibly versatile.
That's truly remarkable flexibility. CRISPR-Cas9 isn't just about cutting and pasting genes, it's this entire programmable platform for interacting with the genome. And we're already seeing this move beyond the lab bench into animal models and even clinical trials.
Yes, the translation has been incredibly rapid. In vivo, applications are progressing quickly. For example, researchers have successfully corrected a fatal hereditary liver disease called tyrosinemia in adult mice by directly delivering the CRISPR-Cas9 components in a donor DNA template to liver cells. Similar successes have been achieved in correcting the mutation causing Duchenne muscular dystrophy in mouse models, sometimes even by direct injection into the germline to create corrected offspring.
In human trials.
Yes, clinical trials using CRISPR-Cas9 edited cells are underway. Early trials, particularly in China and now elsewhere, have involved taking T cells from cancer patients, editing them ex vivo outside the body, using CRISPR-Cas9 to enhance their cancer-fighting abilities, for example, by knocking out genes that inhibit T cell function, expanding these engineered cells, and then infusing them back into the patient. Trials are also exploring in vivo editing for genetic diseases like sickle cell anemia and beta thalassemia. The pace is astounding. If we connect this to the bigger picture, the rapid adoption and incredibly diverse applications of CRISPR-Cas9 really underscore the power of understanding fundamental biological mechanisms, in this case, bacterial immunity, to develop truly transformative technologies that are reshaping medicine and biological research.
It's a stunning example. Now, for many complex immunological questions, especially those involving interactions between different cell types, tissues, and the whole organism's response, you ultimately need to study the immune system within a whole living animal. That brings us to whole animal experimental systems, and the mouse is king here, right?
The mouse is certainly the primary model, yes. While the choice of animal model is always dictated by the specific scientific question, you might need primates to study primate-specific viruses, for instance, and other species like rats, rabbits, or even larger animals are used. The mouse remains the most frequently and arguably most effectively used model for studying the fundamental principles of the mammalian immune system.
Why the mouse?
Several reasons. They're relatively small, easy to handle, and breed quickly. Their genetics are extremely well characterized, arguably better than any mammal besides humans. And crucially, as we'll discuss, there's an incredible toolkit for genetic manipulation available specifically in mice, allowing us to create models that mimic human immune functions and diseases with remarkable fidelity.
But using animals in research obviously raises significant ethical considerations. How is this managed?
That's a critical point. The huge advances in immunology and medicine owe a great debt to animal models, but these advancements come with a profound ethical responsibility. Concerns about animal welfare have led to stringent laws and ethical guidelines governing animal research worldwide.
Like the IACUC in the U.S.
Exactly. In the United States, any institution receiving federal research funds must have an Institutional Animal Care and Use Committee, or IACUC. This committee, which includes veterinarians, scientists, and community members, reviews and must approve all proposed animal experiments, ensuring they meet strict criteria for necessity, minimization of pain and distress, and appropriate care. The USDA enforces compliance. Failure to adhere to these standards can result in loss of funding and the privilege to conduct research. These standards are continually evaluated and updated globally.
And there's a principle of the 3 R's.
Yes, the 3 R's, first proposed by Russell and Birch back in 1959, form the ethical bedrock of animal research. 1. Refine. Continuously improve experimental techniques and housing conditions to minimize any potential pain, suffering, or distress for the animals. 2. Reduce. Use the minimum number of animals necessary to obtain statistically valid and scientifically meaningful results. 3. Replace. Whenever possible, replace the use of live animals with non-animal alternatives such as cell cultures, computer modeling, or in vitro assays. 3. Replace. Researchers are ethically obligated to adhere to these principles.
It's vital work, but the ethical considerations are paramount. It's a constant balance. Now, within mouse models, to make experiments more reproducible and less noisy, immunologists rely heavily on inbred strains. What exactly are those?
Inbred strains are essential for reducing the experimental variation caused by genetic differences between individuals. An inbred mouse strain is created by repeated, systematic brother-sister matings for at least 20 consecutive generations. This intensive inbreeding process leads to animals that are homozygous, have 2 identical copies at virtually all 99% of their genetic loci.
So all mice within an inbred strain are essentially identical twins, genetically speaking.
Yes, they are genetically identical, or syngeneic. This genetic uniformity is incredibly valuable because it means any differences you observe in an experiment between groups of these mice are much more likely to be due to your experimental manipulation rather than underlying genetic variations between individuals. There are hundreds of different inbred mouse strains, commercially available strains like C57BL6, the most common, BALBC, NOD, a model for diabetes, etc., each with well-characterized genetic backgrounds and susceptibilities to different diseases or immune responses.
Okay, so inbred strains are genetically uniform. What about congenic strains? They sound similar but have a specific twist.
Congenic strains are another powerful tool for dissecting gene function. 2 strains are considered congenic if they are genetically identical, except at a single specific genetic locus or a very small, defined chromosomal region.
How are they made?
Typically by repeatedly back-crossing mice carrying a specific genetic difference onto a standard inbred background strain, selecting for the desired genetic difference in each generation. After many back crosses, the resulting strain is essentially identical to the background strain, but it retains that small differential region.
What's the advantage of that?
It allows you to isolate the effect of that single differing genetic region. Any phenotypic differences you observe between the congenic strain and its background partner strain must be due to the genes within that small differential segment. Congenic strains were absolutely critical, for example, in mapping the genes responsible for transplantation rejection to the major histocompatibility complex, MHC, and in dissecting the functions of different MHC subregions.
So, precise genetic tools for controlling the background. Now, what about studying the function of specific cells in vivo? Can you transfer cells between these mice?
Yes, that's done using adoptive transfer experiments. This is a foundational technique in cellular immunology. Because mice within the same inbred strain are syngeneic, you can isolate immune cells, like specific lymphocyte subpopulations, from one mouse, the donor, and inject them intravenously into another mouse of the same strain, the recipient, without causing immune rejection.
What does that let you study?
It allows you to directly assess the in vivo function of the transferred cells. Early adoptive transfer experiments were pivotal. They showed that lymphocytes from an antigen-primed mouse could transfer immunity to an unprimed recipient, proving lymphocytes carry immunological memory. They also demonstrated that both T cells and B cells are required for antibody responses by transferring purified populations.
Can you track the transferred cells?
Yes, you can use donor and recipient mice that differ slightly in non-immunogenic markers, like CD45 allelic variants, that can be detected with specific antibodies. Or, more commonly now, you can label the donor cells with fluorescent dyes, like CFSC, before transfer and track their migration, proliferation, and fate in vivo using flow cytometry or intervital microscopy. Sometimes the recipient mice are irradiated before transfer to eliminate their own lymphocytes, providing a clean slate to study the function of the donor cells without interference.
Creating a customized living system to test cell function. Very powerful. And taking genetic modification to the whole animal level, we have transgenic animals. This is where you introduce entirely new genes, right?
Exactly. Transgenic animals are those that have had foreign DNA, a transgene, stably integrated into their genome. The development of techniques to create transgenic mice back in the early 1980s revolutionized our ability to study gene function in vivo. If the transgene integrates into the germline cells, sperm or eggs, it can be transmitted to offspring like any other gene, allowing the creation of stable transgenic mouse lines.
How is it done? Injecting DNA into eggs.
The most common method is pronuclear microinjection. Cloned DNA containing the transgene is physically injected using a very fine needle directly into the pronucleus, the nucleus of the sperm or egg before they fuse, of a fertilized mouse egg. These manipulated embryos are then surgically transferred into the oviduct of a pseudopregnant surrogate mother mouse.
And some of the resulting pups will carry the transgene.
Yes, typically around 10–30% of the offspring born will have integrated the transgene into their chromosomes, becoming transgenic founders.
Can you control where or when the transgene is expressed?
Yes, that's a crucial aspect. The transgene construct usually includes promoter and enhancer sequences that drive its expression. By using tissue-specific promoters, e.g., a promoter active only in T cells, you can restrict transgene expression to a specific cell type. By using inducible promoters, for example, one activated by a drug like tetracycline or tamoxifen, you can turn the transgene on or off at specific times during development or in response to experimental manipulation.
That gives you spatial and temporal control. But the transgene inserts randomly, right? Could that cause problems?
It does insert randomly into the genome, and that is a significant limitation. Random insertion can disrupt essential endogenous genes, leading to unexpected phenotypes or even lethality. It could also insert into a region of condensed inactive chromatin, resulting in poor or variable transgene expression. This randomness led to the development of more precise methods.
Which brings us to knock-in and knock-out technologies. This is about targeting genes precisely.
Exactly. These technologies overcome the limitations of random transgenesis by using homologous recombination to target genetic modifications to specific, predetermined sites in the mouse genome, usually replacing the endogenous gene.
So what's the difference between knockout and knock-in?
A knockout mouse is one where the normal allele of a specific gene has been replaced or disrupted by a non-functional version. This allows researchers to study the effects of completely losing the function of that gene throughout the animal's development and life. A knock-in mouse is one where the endogenous gene has been replaced by an altered version, perhaps a version carrying a specific human disease mutation, or a version where the coding sequence is followed by a reporter gene like GFP, allowing you to visualize which cells are expressing that gene.
So targeted deletion or targeted replacement modification. This sounds much more precise, but also more complex to achieve. How is it typically done? using those embryonic stem cells.
Yes. The generation of knockout and knock-in mice classically relies on manipulating mouse embryonic stem, ES cells. ES cells are derived from the inner cell mass of an early mouse embryo, the blastocyst. They are pluripotent, meaning they can differentiate into all cell types of the body, and importantly, they can be cultured and genetically manipulated in vitro.
So you modify the ES cells first.
Right. You first design a targeting construct. This is a piece of DNA containing your desired modification, e.g., the disrupted gene for a knockout or the altered gene for a knock-in, flanked by sequences that are homologous, identical to the DNA sequences immediately upstream and downstream of the target gene in the mouse genome.
Those homologous arms guide the recombination.
Exactly. You introduce this targeting construct into the cultured ES cells, usually by electroporation. In a small fraction of cells, homologous recombination will occur between the targeting construct and the endogenous gene locus, replacing the normal gene with your engineered version.
How do you find those rare cells where it worked correctly?
That requires a clever selection strategy. The targeting construct typically includes positive selection markers, like the NER gene, which confers resistance to the drug G418 neomycin. This allows you to kill off any ES cells that didn't take up the DNA at all. Crucially, for selecting against random integration events, the construct often includes a negative selection marker, like the TKHV gene, thymine kinase from herpes simplex virus, located outside the homologous arms.
How does the negative marker work?
The TKHSV gene makes cells sensitive to the drug ganciclovir. If the targeting construct integrates randomly into the genome, the TKHSV gene is likely to be incorporated along with it. However, if integration occurs correctly via homologous recombination at the target site, the TKHSV gene, being outside the homology arms, is lost. So you treat the cells with both G418 kills non-integrants and ganciclovir kills random integrants. Only the ES cells that have undergone the desired homologous recombination event will survive this double selection.
Brilliant. Positive selection for integration, negative selection against random integration. What happens to the selected ES cells?
These correctly targeted ES cells are then injected into a recipient mouse blastocyst, often from a mouse strain with a different coat color. Exim inject black ES cells into a white blastocyst. This manipulated blastocyst is implanted into a pseudopregnant surrogate mother.
And if the ES cells contribute?
The resulting offspring may be chimeric, composed of a mixture of cells derived from the host blastocyst, white, and the injected ES cells, black. A chimeric mouse, for example, with patches of black and white fur, indicates that the engineered ES cells successfully contributed to the developing embryo, including, potentially, the germline.
So you breed the chimeras.
Yes, you breed the chimeric mice. If the engineered ES cells contributed to the germline, sperm or eggs, some of their offspring will inherit the targeted genetic modification in all their cells. These heterozygous offspring can then be interbred to produce homozygous knockout or knock-in mice, which carry the modification on both copies of the chromosome.
That is an incredibly intricate process, but the payoff is huge. Having an animal model where you've precisely altered a specific gene. Amazing control. But you mentioned earlier, sometimes knocking out a gene completely from the start is lethal if it's essential for development. Or maybe you only want to delete a gene in the specific tissue, or only after the animal is born. How do you get that level of conditional control?
That's where another layer of genetic sophistication comes in, most commonly using site-specific recombinase systems, like the KRELOC system. This allows for conditional gene targeting, deleting or activating a gene only in specific cell types, or only at specific times.
KRELOCs sounds like something from bacteria or yeast.
It is. The Cre recombinase enzyme comes from bacteriophage P1. Cre recognizes specific short DNA sequences called LOXP sites, locus of X over P1. When Cre encounters two LOXP sites oriented in the same direction on the same DNA molecule, it catalyzes recombination between them, precisely excising the DNA segment located between the two LOXP sites.
So if you put LOXP sites around a gene, Cre will cut it out.
Exactly. This allows for conditional gene deletion. The strategy involves creating two different lines of mice and breeding them together. First, you create a mouse line where your target gene is floxed, meaning it's flanked by LOXP sites, but is otherwise still functional. This is usually done using ESL technology. Second, you create a separate transgenic mouse line that expresses the Cre recombinase enzyme, but crucially, under the control of tissue-specific promoter. For example, a promoter that is active only in T lymphocytes.
Okay, one mouse has the flox gene, the other makes Cre and T cells. What happens when you breed them?
In the offspring that inherit both the flox gene and the T-cell specific Cre transgene, The Cre enzyme will be expressed only in their T cells. In those T cells, Cre will recognize the LOXP sites flanking the target gene and excise it. The gene will be deleted only in T cells while remaining intact in all other tissues where Cre is not expressed. This allows you to study the function of a gene specifically within a particular cell lineage, even if that gene is essential for overall viability.
That's incredible spatial control. Can you get temporal control too? Turn the deletion on or off when you want?
Yes, this is often achieved using inducible Cre systems. A common approach is to use a fusion protein where Cre recombinase is linked to a mutated estrogen receptor ligand binding domain. This fusion protein normally stays sequestered in the cytoplasm. However, when the animal is treated with the synthetic estrogen analog tamoxifen binds to the receptor domain, causing the Cre fusion protein to translocate into the nucleus, where it can then find the LOXP sites and mediate recombination.
So you control the timing of gene deletion simply by giving the mouse tamoxifen.
Exactly. This allows researchers to delete a gene at a specific developmental stage or only after an immune response has been initiated, for example. The CRELOX system can also be used for conditional gene activation. For instance, you can insert a LOXP-flanked transcriptional STOP cassette upstream of a gene you want to turn on. The gene is initially off. When Cre is expressed, either tissue-specifically or inducibly, it removes the STOP cassette, allowing the gene to be transcribed. This level of conditional control, spatial and temporal, allows investigators to ask incredibly precise questions about gene function in the context of a whole complex organism. What happens if this gene is deleted only in mature B cells and only after they've encountered antigen? The CRELOX system, combined with floxed alleles, provides the tools to answer these sophisticated biological questions.
That is just an astounding level of finesse in manipulating biological systems. We have certainly covered a vast and incredibly sophisticated arsenal of experimental methods today. It's really mind-boggling.
It truly is. From harnessing the remarkable specificity of antibodies for detection, purification, and visualization, to the intricate techniques of cell sorting and dynamic intravital imaging, and finally, to the revolutionary power of genetic manipulation with CRISPR-Cas9 and these sophisticated conditional knockout systems in whole animals. Immunologists have developed and continue to refine an unparalleled toolkit for dissecting the immune system.
And the value proposition here for you, our listener, is immense, we hope. Understanding the principles behind these methods, the why and how, allows you to read the scientific literature in immunology, or even reports about new diagnostics and therapies, far more knowledgeably. You can evaluate the experimental choices made by investigators more critically, appreciate the strengths and limitations of different approaches.
Right. It's about understanding the foundation upon which our knowledge is built.
Exactly. It's really about empowering you to engage with and master this information, like the elite professionals in this field, to understand the logic behind the experiments that drive discovery.
So looking forward, as we continue to push the boundaries of what's possible, combining these powerful techniques in new and synergistic ways, maybe high-parameter site-off with spatial transcriptomics or CRISPR screens with advanced imaging, what new frontiers will be open, and perhaps just as importantly, what new ethical questions might arise as our ability to observe and manipulate complex biological systems becomes ever more profound.
Excellent points to ponder. The journey of discovery in immunology, propelled by these ingenious methods, clearly continues at an accelerating pace. Thank you for taking this deep dive with us today.